Welcome to Chapter 1 of Class 12 Biology, Sexual Reproduction in Flowering Plants. In this fascinating chapter, we'll explore how flowering plants reproduce sexually, creating the incredible diversity of flowers, fruits, and seeds we see around us. Biology tells the story of life on Earth, and while individual organisms inevitably die, species continue through millions of years via reproduction. Sexual reproduction is particularly important because it enables creation of new variants, enhancing survival advantages. The myriads of flowers we enjoy gazing at, the scents and perfumes we swoon over, the rich colors that attract us, all these exist as aids to sexual reproduction. Flowers don't exist only for human enjoyment, they serve crucial reproductive functions that ensure species survival. In this chapter, we'll examine flower structure, pollen development, embryo sac formation, pollination mechanisms, double fertilization, and seed development, the complete reproductive journey in flowering plants. Let's begin with the flower itself, nature's marvel of reproductive engineering. Human beings have had an intimate relationship with flowers since time immemorial. Flowers hold aesthetic, ornamental, social, religious, and cultural value across civilizations. But to a biologist, flowers represent morphological and embryological marvels, the specialized sites of sexual reproduction in flowering plants. A typical flower consists of four main parts: the calyx made of sepals that provide protective covering, the corolla composed of petals that attract pollinators, the androecium, which is the male reproductive part containing stamens, and the gynoecium, the female reproductive part consisting of pistils. The two most important units for sexual reproduction develop in specific parts. The male gametes develop in the anthers of stamens, while the female gametes develop in the ovules within the ovary of the pistil. This specialized structure makes flowers incredibly efficient reproductive organs. Now let's explore what happens before fertilization, the pre-fertilization structures and events. Much before we actually see a flower on a plant, the decision to flower has already been taken. Several hormonal and structural changes initiate the differentiation and development of the floral primordium. Inflorescences form, bearing floral buds that eventually develop into flowers where the male and female reproductive structures differentiate. The androecium, representing the male reproductive organ, consists of stamens. Each stamen has a long slender stalk called the filament and a terminal generally bilobed structure called the anther. A typical angiosperm anther is bilobed with each lobe having two theca, making it ditheceous. It's a four-sided tetragonal structure containing four microsporangia located at the corners. These microsporangia develop further into pollen sacs that extend longitudinally through the anther and become packed with pollen grains. The microsporangium is surrounded by four specialized wall layers that protect and help in pollen release. Let's dive deeper into microsporogenesis and pollen grain development. Microsporogenesis is the process where microspores form from pollen mother cells through meiosis. The cells of the sporogenous tissue in the center of each microsporangium undergo meiotic divisions to form microspore tetrads. Each cell of this tissue is a potential pollen or microspore mother cell. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, thousands of microspores or pollen grains form and are released during anther dehiscence. Pollen grains represent the male gametophytes. If you touch opened anthers of flowers like hibiscus, you'll find yellowish powdery pollen grains deposited on your fingers. These grains show amazing architectural variety in sizes, shapes, colors, and designs when viewed under a microscope. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter, with a prominent two-layered wall. Now let's examine the female reproductive structures, the pistil, Megasporangium, and embryo sac. The gynecium represents the female reproductive part of the flower and may consist of a single pistil or multiple pistils. When there are multiple pistils, they may be fused together or remain free. Each pistil has three essential parts: the stigma that serves as a landing platform for pollen grains, the style which is the elongated slender part beneath the stigma, and the ovary which is the basal bulged part containing ovules. Inside the ovary is the ovarian cavity with placenta, from which arise the megasporangia, commonly called ovules. A typical angiosperm ovule has several key parts: the funicle stalk attaching it to the placenta, the hilum where the ovule body fuses with the funicle, integuments that are protective envelopes, the micropyle opening, the chalaza at the basal part, the nucellus mass of cells with food reserves, and the embryo sac or female gametophyte. The number of ovules varies greatly among species. Let's explore megasporogenesis and embryo sac development in detail. Megasporogenesis is the process where megaspores form from the megaspore mother cell. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. This MMC is a large cell containing dense cytoplasm and a prominent nucleus that undergoes meiosis. In the majority of flowering plants, one of the resulting megaspores becomes functional while the other three degenerate. Only the functional megaspore develops into the female gametophyte or embryo sac through a process called monosporic development. The embryo sac forms through several stages. First, the nucleus of the functional megaspore divides mitotically to form two nuclei that move to opposite poles, creating the two-nucleate embryo sac. Two more sequential meiotic nuclear divisions result in 4 nucleate and then 8 nucleate stages. These divisions are free nuclear, meaning nuclear divisions occur without immediate cell wall formation. After the 8 nucleate stage, cell walls form, organizing the typical female gametophyte. Now let's discuss pollination, the crucial process of pollen transfer. Since both male and female gametes in flowering plants are non-motile, they need to be brought together for fertilization to occur. Pollination is the mechanism that achieves this objective. It's the transfer of pollen grains from the anther to the stigma of a pistil. Flowering plants have evolved an amazing array of adaptations to achieve pollination using external agents. There are 3 main types of pollination based on the pollen source. Autogamy is self-pollination within the same flower, requiring synchrony between pollen release and stigma receptivity, plus close proximity between anthers and stigma. Geitonogamy involves transfer between different flowers of the same plant. While functionally cross-pollination, it's genetically similar to autogamy. Xenogamy is transfer between flowers of different plants, and this is the only type that brings genetically different pollen to the stigma. Some plants produce cleistogamous flowers that don't open at all, ensuring autogamy and assured seed set even without pollinators. Let's examine the various agents that help in pollination. Plants use both abiotic and biotic agents to achieve pollination. The abiotic agents are wind and water, while the main biotic agent is animals. Interestingly, the majority of plants use biotic agents, with only a small proportion relying on abiotic methods. Wind pollination is more common among abiotic methods and requires specific adaptations. Pollen grains must be light and non-sticky for wind transport. Flowers need well-exposed stamens for easy pollen dispersal, and they often have feathery stigmas to trap airborne pollen. Wind-pollinated flowers typically have single ovules per ovary and numerous flowers packed into inflorescences, corn being a familiar example. Water pollination is quite rare in flowering plants, limited to about 30 genera, mostly monocotyledons. In contrast to higher plants, water is a regular transport mode for male gametes in lower plant groups like algae, bryophytes and pteridophytes. Biotic pollination involves a wide range of animals including bees, butterflies, birds, bats, and even some reptiles. Now let's explore the fascinating outbreeding devices that plants have evolved. Majority of flowering plants produce hermaphrodite flowers where pollen grains are likely to come in contact with the stigma of the same flower. However, continued self-pollination results in inbreeding depression, reduced biological fitness due to inbreeding. To counter this, flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination, which increases genetic diversity. One common device is dichogamy, the non-synchronization of pollen release and stigma receptivity. This can be protandry where anthers mature before the stigma, or protogeny where the stigma matures first. Another device is hercogamy, the spatial separation of anthers and stigma within the same flower to prevent self-pollination. The third important device is self-incompatibility, a genetic mechanism that prevents self-pollen from fertilizing ovules by inhibiting pollen germination or pollen tube growth in the pistil. Additionally, some plants produce unisexual flowers to prevent self-pollination. Let's understand the crucial process of pollen-pistil interaction. It's important to note that pollination doesn't guarantee the transfer of the right type of pollen. Often, pollen of the wrong type, either from other species or from the same plant if it's self-incompatible, also lands on the stigma. The pistil has the remarkable ability to recognize whether pollen is compatible or incompatible. If it's the right type, the pistil accepts the pollen and promotes post-pollination events leading to fertilization. If it's the wrong type, the pistil rejects it by preventing pollen germination or pollen tube growth. This ability to recognize pollen followed by acceptance or rejection results from a continuous dialogue between the pollen grain and the pistil, mediated by chemical components. Only in recent years have botanists been able to identify some of these pollen and pistil components and their interactions. Following compatible pollination, the pollen grain germinates on the stigma, producing a pollen tube that grows through the tissues to reach the ovary and ultimately the embryo sac. Now we come to one of the most unique events in flowering plants, double fertilization. After the pollen tube enters one of the synergids, it releases two male gametes into the synergid's cytoplasm. One of these male gametes moves toward the egg cell and fuses with its nucleus, completing syngamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves toward the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus. Since this involves the fusion of three haploid nuclei, it's termed triple fusion. The occurrence of these two types of fusions, syngamy and triple fusion, in the same embryo sac is called double fertilization, an event unique to flowering plants. After these fusions, the central cell becomes the primary endosperm cell and develops into the endosperm, while the zygote develops into an embryo. This dual fertilization ensures that both the embryo and its food supply develop simultaneously. Let's examine what happens after fertilization, the post-fertilization events. Following double fertilization, several important developmental processes occur: endosperm development, embryo development, maturation of ovules into seeds, and transformation of the ovary into fruit. These are collectively termed post-fertilization events. Endosperm development always precedes embryo development, an adaptation to provide assured nutrition to the developing embryo. The primary endosperm cell divides repeatedly, forming triploid endosperm tissue. In the most common type, the pen undergoes successive nuclear divisions to give rise to free nuclei, a stage called free nuclear endosperm. Subsequently, cell wall formation occurs, and the endosperm becomes cellular. The embryo develops at the micropolar end of the embryo sac where the zygote is situated. Most zygotes divide only after a certain amount of endosperm has formed. Though seeds differ greatly, the early stages of embryo development are similar in both monocotyledons and dicotyledons. Now let's focus on seed formation, the final product of sexual reproduction. In angiosperms, the seed is essentially a fertilized ovule formed inside fruits. A typical seed consists of seed coats, cotyledons, and an embryo axis. The cotyledons of the embryo are generally thick and swollen due to storage of food reserves, especially in legumes. Mature seeds are classified based on their endosperm status. Non-albuminous or X-albuminous seeds have no residual endosperm as it's completely consumed during embryo development, while albuminous seeds retain part of the endosperm as it's not completely used up. Occasionally, in some seeds like black pepper and beet, remnants of nucellus persist as perisperm. The integuments of ovules harden into tough protective seed coats, with the micropyle remaining as a small pore for oxygen and water entry during germination. As seeds mature, their water content reduces significantly, and they may enter dormancy, a state of inactivity that allows survival in unfavorable conditions. Let's explore fruit development and the different types of fruits. As ovules mature into seeds, the ovary simultaneously develops into a fruit. The wall of the ovary becomes the fruit wall called pericarp. Fruits can be classified in several ways. Based on texture, they can be fleshy like guava, orange, and mango, or dry like groundnut and mustard. Based on origin, we have true fruits that develop only from the ovary, and false fruits where other floral parts contribute to fruit formation. Based on fertilization, there are normal fruits that result from fertilization, and parthenocarpic fruits that develop without fertilization. In most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in some species like apple, strawberry, and cashew, the thalamus also contributes to fruit formation, making them false fruits. Parthenocarpy can occur naturally or be induced through application of growth hormones, producing commercially important seedless fruits like bananas. Finally, let's examine two special reproductive mechanisms, apomixis and polyembryony. Although seeds are generally products of fertilization, some flowering plants have evolved apomixis, a mechanism to produce seeds without fertilization. Apomixis is essentially a form of asexual reproduction that mimics sexual reproduction. There are several ways apomictic seeds develop. In some species, the diploid egg cell forms without reduction division and develops into the embryo without fertilization. More commonly, as in many citrus and mango varieties, some nuclear cells surrounding the embryo sac start dividing, protrude into the embryo sac, and develop into embryos. This leads to polyembryony, the occurrence of more than one embryo in a seed. Apomixis has tremendous importance in agriculture, especially for hybrid crops. Normally, hybrid seeds have to be produced every year because seeds collected from hybrids don't maintain hybrid characters. If hybrids are made into apomix, there's no segregation in the progeny, allowing farmers to reuse seeds year after year, significantly reducing costs.